Although reports of original endogenous biochemistry from the Cenozoic, more recent periods are considered, the focus of the review is on claims of original biochemistry from the Mesozoic, Paleozoic, and lower levels, which are more difficult to explain. All right, let's go back to the geologic column again. Not so much the rocks themselves, but the time units that the evolutionists impose on those rocks. When they're looking at rocks, they're not just looking at rock. In their mind, they're looking at a deposit during a certain period of time. They divide most of the fossil record into these three main eras of time. The most recent being the Cenozoic. The word Cenozoic is actually Greek for recent life. Cenozoic. Mesozoic is middle life. And Paleozoic is old life. Okay? Cenozoic, they would date that, supposedly, between 65 million years or so to the present. Okay? So we're living, supposedly, in the age of mammals, which is the Cenozoic era. Prior to that, 65 million years or so, back to about 225 million years, as far as their view of Earth history, would be the Mesozoic, the middle life era, which is divided into three periods. This is called the Age of Reptiles, which precedes the Age of Mammals, and is divided into the Triassic Period, the Jurassic Period, and the Cretaceous Period. Okay? Most people are familiar with the word Jurassic because of the Jurassic Park movies, Jurassic World movies. Jurassic was basically the heyday, supposedly, of the dinosaur time period. But then that's followed by the Cretaceous period. Creta is the Latin word for chalk. And we have a lot of calcium carbonate, limestone deposits in the Cretaceous. The rock here in the Glen area, all the way down to Biloxi, down the I-35 corridor, down to Austin and farther south, is basically limestone. It's the Glen Rose limestone. It's found on at least 6 continents around the globe. It is a worldwide unit here. And it's the unit that our dinosaur and human footprints are in. And so they're dated as Cretaceous, which is part of the Mesozoic. And then prior to all that, we have the Paleozoic, which supposedly are the amphibians, the fish, the simple invertebrates, all the way back to the first life that supposedly evolved from non-life. That's another whole issue, right? And so this is why these areas, the Mesozoic, Paleozoic, and lower levels are more difficult to explain, is because, in their view, these rocks are much, much older. So what's organic tissue doing in those? Institute for Creation Research scientist Jeffrey Tompkins, PhD, and he's famous for doing research on the DNA of chimpanzees and humans, among other things. He's a great writer, great researcher, but he's really made a name for himself in debunking the myth that chimpanzee DNA is 98 to 99% similar to human DNA. That's in the textbooks. Our kids are learning that. It is totally false. He's gone back, he's looked at how those figures come about, the research behind all of that, and he says basically the closest chimpanzees can be to humans is about 84%. And you may think, well, that's still 84%. Well, the 16% difference is 16% of our whole genetic code, which is about 3.2, 3.3 billion nucleotide base pairs in the DNA. We're talking a world apart from chimpanzees. Physically, we have 2 arms, we have 2 legs, we're bilaterally symmetrical. We probably eat some of the same foods they do, those kind of things. And so you would expect some similarity in the design, in the engineering of the organisms. But to say that we're only 1 to 2% different than a chimpanzee, that is totally false. And Jeffrey Tompkins' research bears that out. He wrote this: soft tissues and decay-sensitive biomolecules that are still intact and not degraded shouldn't exist in fossils that are supposedly millions of years old, but they do. The most famous case of this evolutionary enigma was the discovery of soft, stretchy tissue in the bones of a T-Rex, along with visible blood vessels, blood cells inside the vessels, and bone cells, osteocytes, with delicate finger-like projections, right, called osteocytes. And here in the picture, that white mass, more or less in the center, is a Haversian canal. Haversian canals are in pretty complicated organisms that have blood vessels and nerves that run through the bone. And that's what you're seeing there is one of the passageways, cross-section of a passageway, where either a nerve or a blood vessel would go to basically service that bone. The little black specks that look like little watermelon seeds everywhere, those are the osteocytes. Okay? And remember, bone is a living tissue. It's just more calcified than most tissue. Collagen proteins were also found in the T-Rex bones. Collagen is a very important protein molecule. It helps to actually give structure and support for bone material and other tissues. Found in T-Rex bones, there are two types of collagen. There's a type 1 collagen, which is bone collagen, and then there's type 2 collagen, which is cartilage collagen, protein that makes up cartilage. Some of these biomolecules have experimental ranges of how long they can last, and then the evolutionists go and then extrapolate a maximum age for that as well, based on experimental evidence. Bone collagen is about 30,000 years. They extrapolate that to 900,000 years. 30,000 to 900,000 years. Can you still see there's a problem there, though? 900,000 years is less than 1 million. And yet, some of the bones that they're supposedly dating are between 65 and 70 million years. Okay, so we got a problem with that. Bone collagen would certainly last longer than cartilage collagen, which has a duration of far less than 30,000 years. Mitochondrial DNA, we found that in some of these fossils. That DNA has a half-life, the time it takes for half of it to decay, of 521 years. So if after 521 years, half of it should be gone. And then another 521 years, half of that half should be gone. So we'll go from 50% to 25% to 12.5% to 6.25%. You can kind of see taking half of a half of a half. After about 7 or 10 half-lives, you're down to less than 1% of the original material. So that means that the mitochondrial DNA, if that's the protein, if that's the molecule we're looking at, that has a duration of less than 10,000 years. Less than 10,000, and that's at the most.